So as means of an introduction, this series began back in 2018 between Dr. Peter Aldiss and I, where we were encouraging early career researchers to really disseminate their research to wider audiences. So after symposiums at the European Congress of Obesity and then Experimental Biology, and accumulating in a one-day seminar at Physiology 2019, we decided to take this series online to hopefully negate some of the consequences that we're all experiencing due to the global pandemic. Throughout, the focus has been not only the promotion of early career researchers, but offering them the opportunity to connect, as we will ultimately be the next generation of leaders within the field. Now, before I hand over to Pardeep, who's co-chairing the session today, a PhD student at the University of Nottingham, I just wanted to run through some brief housekeeping notes. Our speakers today will deliver a 20-minute talk each, and after both of those talks, we'll have a 15-minute session for question and answers, and you can submit those through the button below. You have the option to submit these questions anonymously, or if you leave your name and your institution, both Pardeep and I can read those out as part of your questions. Please note that today's session is being recorded and will be made available subsequently on the Physiological Society's YouTube channel. I'd just like to thank both of our speakers for their talks today and hope for an engaged session. Thank you very much. And Pardeep, if I could hand over to you.
Yeah, thank you very much, Joe. Hello, everyone. Warm welcome from me. So kicking off today, we have Dr. Sarah Turpin-Nolan from the Monash University in Australia. Sarah is a research fellow there now, and she investigates the role of ceramides in metabolic disease in the GI tract and lymph and circulatory systems. And during her PhD, she looked at the role of specific ceramide species in muscle and the liver. So Sarah's talk is entitled The Contribution of Ceramides to Metabolic Dysfunction. Over to you, Sarah. Thank you very much.
Thank you for that introduction. I'm just sharing my screen now and I'm hoping everyone can see that. So first of all, thank you for the invitation to present today. It's actually midnight here in Melbourne, Australia. So I hope everything goes to plan here, and I obviously won't fall asleep because I'm quite wired up to present to you today. So just a bit of background about myself. I completed my honours and PhD with Professor Matthew Watt at the University of Melbourne and also Monash University. It was after that I then moved over to Cologne in Germany, and I worked in the laboratory of Professor Jens Bruning when we were at the University of Cologne, where I was supported by Alexander von Humboldt Fellowship. He then became the director of a Max Planck Institute, which was renamed Max Planck Institute for Metabolism Research. And during that time and the work that I'll present to you today, we had a successful collaboration that's still ongoing with Sanofi. And I also received a grant to try and undertake some independent research to set myself up for my future career. Obviously, I've had some career breaks in that time, having kids and relocating back to Melbourne, Australia. And currently, I'm a research fellow in the laboratory of Professor Mark Fabreo at Monash University. We're in the Institute of Pharmaceutical Sciences. And within Mark's lab, I'm now working on his next generation of designer cytokines. Some of you might be aware that he published last year on his designer cytokine IC7. We're now looking at the next generation of those cytokines. And I'm also undertaking my own independent research, which I'll talk to you as well later. So how do ceramides influence metabolism during the metabolic state of obesity? And to guide you through this, I'll give you a bit of background and then take you through two of the publications during my postdoc time in Germany. So the lipid physiology, we all really know about triacylglycerol, diacylglycerol, and fatty acids, but there's also another family of lipids which I've become quite involved with, which is the sphingolipid family. And sphingolipids have a sphingonine backbone, and they also, when they produce ceramides, have a fatty acid attached to them. And ceramide synthases are the enzymes, which are involved in attaching these fatty acids to the sphingolipid backbone to produce ceramides of different chain lengths. And these are then referred to as the fatty acid chain length. Ceramide metabolism occurs throughout this pathway. What we like to say is that there's this de novo arm in pink, which is predominantly the arm that's focused upon during metabolic disease. We feel that this is where the high flux of fatty acids come into the pathway to ultimately produce ceramides, which is one of the central lipids in this pathway. As you can see, there's a lot of other arms which recycle these ceramides into other sphingolipid forms, but what I've been focusing on is the role of ceramide synthases, which, as I said before, incorporate these fatty acids to produce dihydroceramides, which become desaturated to ultimately result in ceramides. And what we love about these ceramides and the ceramide synthases is that the ceramide synthases are highly specific. And as you can see in the pink diagram here, there's 6 ceramide synthase enzymes, and each one is highly specific in its selectivity for the addition of the fatty acid to the sphingolipid backbone. So ceramide synthase 1 incorporates a C18 or C20 fatty acids. Ceramide synthase 5 and 6 incorporate C16 fatty acids. Ceramide synthase 2 is focusing on more very long chains, so C20 and above. And so what we believed was that in metabolic disorders, could we try and manipulate ceramides of specific chain lengths rather than try and shut down global ceramide metabolism, which had been associated with detrimental effects, especially in animals. And so what we set out to do was to try and identify, is there a specific ceramide synthase that might be able to do that? However, on our travels, we realized it's not that simple. As you can see, the expression of ceramide synthases differs greatly between different tissues. And this is just a representation of that. Obviously, for metabolic disease, we can't just target one ceramide synthase. It needs to be something specific to the organ that you're obviously trying to target. And so what I want to show you now is just a summary of the research that's come out in the last 10 years using genetic mouse models to try and study ceramide synthases. And it hasn't really been until these models have been published that we've actually had an idea of what each might be responsible for. And so up the top, I have the conventional knockouts or whole body knockouts. And you can see ceramide synthase 1 is quite actively involved in the development of the cerebellum. It's also involved in CNS function and some metabolic diseases. Ceramide synthase 2 is involved in hepatocellular carcinomas. It also has a major function in the production of myelin around neurons and type 2 diabetes. Ceramide synthase 3 and 4 are not as metabolic as the others, you might say. So there's ceramide synthase 3 is heavily involved in the production of sperm and keratinocyte differentiation and skin barrier function. Ceramide synthase 4, if you don't have that, you result in a lot of hair loss. And ceramide synthase 5 and 6 have been recently shown to be quite heavily involved in diet-induced obesity, type 2 diabetes, and potentially in behavior attitudes. And what I show below the line is the conditional knockouts of these animals. And so the work that I'll show you today is now in the bold colors. And this is all the work that we performed in Germany with Professor Jens Bruning, and that's what we've been publishing. So I'll take you through those publications now. And I've tried to use it to tell a story of where I'm now getting to with my own independent projects. So to take you right back to the beginning, when I joined Jens's lab in 2010, we were already investigating which would be the best ceramide synthase to target in models of obesity. And so what we'd found with a collaboration with Matthias Bluer was that in human patients, we found that the BMI strongly correlated in white adipose tissue with ceramide synthase 6. And so we went on to study that using genetic models in the mice, where we would feed them a high-fat diet. And as you can see, when we knock out ceramide synthase 6, which is responsible for C16 ceramides, we had quite a specific reduction in only C16 ceramides. We thought we would get a compensation or changes across the board, but what was really nice and showed that using genetic mouse models is that targeting ceramide synthases can be quite specific in trying to manipulate specific ceramide chain lengths. And so in the white and brown adipose tissue and the liver, we were able to selectively reduce C16 ceramides. However, we didn't see this change in skeletal muscle, and I'll come back to that at a later stage. Whilst we'd been able to just manipulate the C16 ceramides in these tissues, this led to a prevention in diet-induced obesity, which resulted in improvements in glucose metabolism. And so this is now summarizing the paper that we had. So in our whole body knockout, we were able to find that we had reduction in fat mass, reduction in adipocyte size in white adipose tissue. We also had less lipid accumulation in the brown adipose tissue. And when we isolated the brown adipocytes specifically from our animals and performed mitochondrial experiments, we were able to show that using a C16 fatty acid substrate, the oxygen consumption of these mitochondria in these brown adipocytes that were isolated had a greater propensity for using oxygen. So indicating that there was a potential that beta oxidation was increased in these animals. However, when a glucose substrate was given, this was not the case. So it really showed us that the brown adipocytes were using lipid as their main fuel source. We also looked in the liver and found that using primary hepatocytes isolated from our knockout animals, we had a significant increase in palmitate oxidation rates, but also an increase in the efficiency of beta oxidation in the livers of these animals. And so to utilize the technology that Jens Bruning has harnessed, which is genetic mouse models, we went on a search trying to find and pinpoint the exact tissue where we thought there might be a more predominant role of C16 ceramides affecting metabolism. And so what we looked at originally was ceramide synthase 6 in the brown adipose tissue, as we had seen this increase in beta oxidation there. And so we generated ceramide synthase brown adipose tissue knockout animals. These animals, I must say, had not 100% deletion in their brown adipose tissue, more like 48%. So the results I'm showing you are basically, if you think about it in physiological terms, is one sort of side of the brown adipose depot actually has the deletion. And so these results we thought were quite remarkable due to that. We had an increase in energy expenditure like we had in our whole body knockouts. We again saw this increase in the mitochondrial oxygen consumption when given a fatty acid substrate, C16 fatty acid substrate, as opposed to the glucose substrate. And we also saw a significant increase in glucose tolerance. However, these results didn't really recapitulate the exact phenotype we saw in the whole body knockout. So we believed that there must be multiple tissues at play in our animals. And so we went back to the liver, as obviously it's a nice big organ. And we had seen the rates of beta oxidation change in the liver as well. So we generated ceramide synthase 6 liver-specific knockouts to try and isolate the role of the liver and what C16 ceramides are doing there. We could show that again, we had this really nice reduction solely in C16 ceramides in the liver. We didn't see the vast improvement in body weight on a high-fat diet, but we did see some tendency that was significant towards the end of their lifespan that we cut short. And we did see this improvement in glucose tolerance. So in a nutshell, this suggested to us that C16 ceramides in both the brown adipocytes and the liver really can influence beta oxidation in the animals. And that's potentially what was driving the body weight body weight gain in obese animals. And so at the same time, we were also looking at C16 ceramides, but from the perspective of ceramide synthase 5. And so we didn't find any differences in these animals, and that sort of perplexed us a little bit, because if you're getting a reduction in C16 ceramides, why aren't we seeing the same phenotype in both animals, in ceramide synthase 5 knockouts as well as ceramide synthase 6 knockouts? And so this work was then taken over by a PhD student in the lab, Philip Hammerschmidt, who has recently published this work and taken on the results that we had and utilized some beautiful sphingolipid click chemistry and additional mouse models. And just to summarize, because it's a great paper to read, he found that ceramide synthase 6 is expressed on the mitochondria in the liver, and C16 ceramides are able to be produced in mitochondrial fractions. And during high-fat diet, it's these C16 ceramides derived from ceramide synthase 6 that are driving mitochondrial fragmentation, systemic insulin resistance, and non-alcoholic liver steatosis. And so when he looked at the high-fat diet animals, that's what he could see. We also saw it in ceramide synthase 5 whole body knockout animals. But if you delete ceramide synthase 6, you improve all these. So that's something that was continued on after I left. So I wanted to bring it back to the muscle aspect, because as I was going through the early stages of my career, I was doing experiments in myotubes, immortalized cell lines, putting palmitate on those, and then looking at lipid regulation or glucose regulation in those. So it was really something that I questioned, was that why aren't the C16 ceramides negatively regulating glucose metabolism in skeletal muscle? And so as I was puzzled by this, I thought this is actually something worth exploring. And so I generated ceramide synthase 6 skeletal muscle knockouts using the NCK-CRE that Jens Bruning had generated himself in Ronald Kahn's lab. And we saw nothing basically, and we couldn't reduce C16 ceramides like in the whole body ceramide synthase knockout in skeletal muscle. So then I thought, well, maybe ceramide synthase 5 might be producing the C16 ceramides in the muscle. So I made a ceramide synthase 5 and 6 skeletal muscle knockout. And again, you can see the C16 ceramides did not reduce at all, even with double deletion of the 2 genes that are specific for the enzymes to produce that. As we didn't see any deletion, there was no surprise that we didn't see any metabolic phenotype in these animals with regards to body weight or even glucose tolerance. But what was more important was that we realized the C16 ceramides are actually not the predominant species in muscle, it's C18 ceramides. And there was quite a number of papers coming out at that time looking in human studies that were also showing in skeletal muscle, C18 ceramides are the predominant species, and that was more what we should target. And so to summarize this manuscript, we went after ceramide synthase 1 in skeletal muscle. We found that the animals had an improvement in glucose tolerance. So we followed that, and using the hyperinsulinemic euglycemic clamp, we were able to show an improvement in the glucose infusion rate in animals that had ceramide synthase 1 deleted solely from skeletal muscle. This resulted in an 80% decrease in C18 ceramides only. So we didn't change triacylglycerol. There was no effect on diacylglycerol species, only C18 ceramides. So this to us was quite remarkable. We couldn't, unfortunately, pinpoint it down to the skeletal muscle specifically itself due to high variation. But what we did find, which we thought was surprising, was that we had an improvement or a reduction of hepatic glucose production in our muscle-specific knockouts during the steady state. So this suggested to us, is there a circulating factor that could be going to the liver to help improve it during glucose metabolism? And so one suggestion had been to us that could muscles release C18 ceramides into the circulation? And is that what then was going to the liver? So could we have had a reduction in C18 ceramides in the circulation, which was then improving hepatic health? Unfortunately, that was not the case, but that was quite interesting to find out that skeletal muscle doesn't seem to produce ceramides to be released into the circulation, as we did not see any difference at all in C18 ceramides. But what we went along to find was that one of the new sort of candidates on the block, FGF21, was increased in gene expression in our knockout animal muscles. We also had an increase in protein expression only in the muscle. We did not see any difference in expression in the liver. And quite substantially, we had a significant increase in FGF21 in the serum. And so when we added a anti-FGF21 neutralizing antibody into our ceramide synthase 1 skeletal muscle knockouts that had improved glucose tolerance, which is the black diamonds down here, we could see that when we added this antibody, the yellow diamonds, we lost this improvement, suggesting that FGF21 is one of the mechanisms which was improving our glucose tolerance in these animals. And so what I wanted to talk to you now is about some of the new aspects of ceramide research that I think are missing in the field at the moment. Obviously, the work I've presented to you so far is published, and that I've now moved on to different labs. One thing that I've identified, or that's come up to me as being quite strange, is that as we've moved with the times for ceramide research, we've gone from looking at total ceramide levels to now specific ceramide species, to these ceramide species being associated with a specific ceramide synthase, to with even looking at that within specific organelles within the cell of different tissues. But during our time of technology, I guess you would call it, and research, our ceramide signaling pathway and how it affects the insulin signaling pathway has not changed at all. And so my current question is our model of ceramide-induced insulin resistance now correct at this point in time? And the reason that I'm asking this is because using our ceramide synthase 1 skeletal muscle knockout, we went after, obviously, in a very detailed manner, trying to identify these signaling intermediates to show that we had the improvements in them. And that's why we had improved sort of glucose metabolism in our animals. We performed Western blots in a number of different ways. As you can see here, this is using an IV approach with tissues taken out after 5 minutes of insulin injection into the IV system. We did not see any difference in phospho-AKT levels at all. We also performed Western blots in animals that were injected with insulin IP and tissues removed 30 minutes later. You can see quite clearly there's no difference between our control animals and our knockout animals. We also performed Western blots in our hyperinsulinemic euglycemic clamp animals. And again, we saw no difference, which, knowing that we have such improvements in these animals in glucose metabolism, it really puzzled me that we weren't seeing this predicted improvement in AKT phosphorylation. We also looked at Pkc isoforms, which you can see here. There was no difference in any of the Pkc isoforms we went after, and we performed the PP2A activity assay as well. And we also didn't see any changes. And so one reason that this signaling pathway for potentially C18 ceramides might be out of date is because a lot of the data trying to look at this was done in cell culture systems. And so one aspect I'm now sort of chasing, or want to also bring to your attention in this seminar series, is that skeletal muscle myotubes, especially C2, C12s or L6s, which are quite commonly used for lipid metabolism, actually have a proportionate swing to C16 ceramide preference. And even by stimulating those cells with a palmitate, a C16 fatty acid, or a C18 fatty acid being stearate, we can't drive C18 production in these cell lines. So these are the same graph, just stretching the axis to show you that C16 ceramides, no matter what substrate you give, are the predominant ceramide within this cell line. And in talks with Anthony Don from the University of Sydney, he had the paper come out with the ceramide synthase 1 inhibitor. He also found it hard that we couldn't go back to look at mechanisms of signaling in any cell sort of culture, because they had also taken primary myocytes from animals and differentiated those into myotubes. But they still, even using primary myotubes, had a greater proportion of C16 ceramides than 18 ceramides, which doesn't represent what is actually occurring in the animal. And even in undifferentiated primary myoblasts, they also saw a different pattern of ceramides. So this is just something that we're following up on and trying to maybe bring to people's attention. And so finally, this is a new avenue of research which I'm trying to address and get independent funding for, is which metabolic organs are actually producing and exporting ceramides. And so we know that the liver can produce ceramides or package them within LDL lipoproteins. I've hopefully convinced you that the skeletal muscle does not produce any ceramides for the circulation. And Scott Summers is actively looking at whether white adipocytes themselves can also release ceramides into the circulation. However, there's one organ, which is the gut, which also appears that it may be able to generate ceramides and release those. So what I'll show you in this last slide is that during a high-fat diet, if we isolate the epithelial cells of the intestine, we can see that we do get increases with a high-fat diet in ceramide species. And this is throughout sort of the intestine lengths. But more importantly, when we do a very crude experiment, which we look at C16 ceramides in the serum, we have control animals. These are all fed chow diet. So I'm not even trying to push the system to generate ceramides, but we have fasted animals, which is our baseline. If we refeed these animals for 2 hours with chow diet, we see this increase in C16 ceramides. And this is theoretically meant to represent what you see that's coming from the intestine. And so using ceramide synthase 6 intestinal epithelial cell knockouts, we see when we fast the animals, we have less than our controls. But when we refeed them again for 2 hours with a chow diet, we don't see this increase, which would be attributed to the intestine. Excuse me. So this is something that I'm now trying to focus on. I've been very lucky that within our institute, we've got Dr. Nelly Travascus, which has shown that in lymph coming directly from the intestine, we do see ceramides in there. And so together we're trying to now target this as an independent experiment. So just to summarize, as you know, some of the work has been published. So I'll skip the first sort of 3 as they've been published for some time now. But what I really wanted to highlight today is that skeletal muscle cultures, immortalized or primary cells, may not be the best models to study ceramide metabolism or even lipid-mediated insulin resistance. Best to go back to animal models that have a complete system intact, and that ceramides are present in the gut epithelial cells and in lymph. And that could be a way that they're entering the body and circulating to the liver. And finally, I would just like to acknowledge, obviously, Professor Mark Fabreo, the lab that I'm in, collaborators, and the labs that I've been a part of over the last 10 to 15 years. So thank you very much for having me, and I'll hand back over to Joe.
Thank you very much, Sarah, for a great talk. It's very interesting. We'll field questions at the end and move on swiftly to our next speaker, who is Dr. Tara Mcdonald. Tara completed her PhD in Canada at the University of Guelph, where she investigated the role of interleukin-6 and estrogen on glucose metabolism. She's subsequently taken up a postdoctoral fellowship at the Joslin Diabetes Center in Sarah Lessard's lab, where she's looking to investigate the interaction between hyperglycemia and the metabolic and physiological responses to aerobic exercise training. And Tara's talk is entitled Mechanisms Underlying Hyperglycemia and Low Exercise Capacity. Over to you, Tara. Thank you very much.
Thank you very much for the introduction. I'm just going to share my screen now and hope that it works. Let's see. Okay, so hopefully that's all right for everyone to see. Yeah, thanks very much. And thank you to the Physiological Society and the organizers for having us today, especially stretched across so many time zones. I have to say, I think I'm the lucky one because it's just after 9 a.m. here. So I got the, the fortunate time slot. But I'll be talking this morning about some work we've been doing for the last, I guess, almost 4 years at this point. I moved to the US for my postdoc in 2016. And we just recently published a manuscript this summer, looking at the mechanisms that underlie low exercise capacity during hyperglycemia. And we focused very heavily on muscle. So I've chosen today to kind of just walk you through the data in this study, first starting with the clinical relationships that link glycemia and exercise capacity in humans, which served as the foundation for a lot of our work going forward. I'll then get into some in vivo model development that we undertook to answer some of the questions that we published in this manuscript. And then I'll go through the in vivo and muscle adaptations to exercise and exercise training, including muscle signaling. Then towards the end, I'll just take a quick second to talk about the mechanisms that we propose that link hyperglycemia to aerobic exercise adaptations and some next steps that I think would be of interest for the field. So probably no surprise to this group. I'm sure a lot of people are familiar that there are many benefits of aerobic exercise. And so repeated bouts of aerobic exercise or aerobic training through activity like running, swimming, and cycling can lead to training adaptations across many, many systems. So these include metabolic improvements in indices like glucose tolerance and insulin sensitivity. We also tend to see mental health, sleep, and cognitive improvements, which is obviously good. Weight management and longevity as well. And I've got increased exercise capacity highlighted at the bottom because this has been a large focus for my prior training and also this current project that I'm presenting now. And just to get everyone on the same page, I wanted to make a note that exercise capacity can also be noted as cardiorespiratory fitness. It is expressed as a distance or time if you're talking about rodents, or VO2 max for humans and preclinical models as well. So all of these will kind of be used interchangeably depending on what the data outcome is today. So we also know the most effective way to increase VO2 max or capacity is by aerobic training. So of course, this is the seminal study from Claude Bouchard, the Heritage study, where they enrolled subjects and had them complete 20 weeks of aerobic training 3 times per week at 50 to 75% of their VO2 max. And what they did was measure VO2 max before and after training and plot the individual response in a waterfall. So we can see the average is around 400 milliliters per minute. In this study, of course, we have a group of typical high responders and also low responders as well. And so this has also been sort of an interesting area to work in, given the attention both physiologically and statistically to this low response phenotype. But I think at this point, there's many papers that show that in response to the same training stimulus, there is a group that has a lower response than average. And what's interesting is that this response to training is highly variable. So this delta VO2 max is a highly complex trait. It's attributed now to about 50% genetics or 50% environment. So basically there's a component of VO2 max that's inherited and another that can be trained. And so in a separate subset of clinical work, this is from Thomas Solomon and John Kerwin's lab, what they show here is that VO2 max actually has a really strong association with poor glycemic control. So if we look at HbA1c, in the pre-diabetic range, we can see that VO2 max is quite low. Conversely, when HbA1c is down towards more of a normal range, VO2 max is much higher. And to mirror or reflect this, when we look at SIOGTT, which is an index of insulin sensitivity, when this is poor, VO2 max tends to be low as well. So this is just to show you that there's a striking inverse relationship between high blood glucose, many different indices, and exercise capacity in many clinical papers. So this begs the question that low VO2 max may have a causal underlying mechanism that's due to hyperglycemia, at least in humans. And since this webinar series is focused on obesity, I wanted to also show you this data as well. So achieving high exercise capacity, either genetically if you're lucky, or through exercise training, protects against mortality. So in our group of healthy humans here with exercise capacity is shown in METs in this case, as exercise capacity improves, the relative risk of death is quite a bit lower. And actually what I found really interesting is that this phenotype is conserved regardless of BMI. So you can see in the overweight subjects in this study, the relative risk of death is substantially lower when VO2 max or exercise capacity is high, and that's also the case in obesity as well. So this benefit of improving your VO2 max occurs even in the presence of other risk factors that we know contribute to disease, including smoking, age, and BMI. So these clinical studies taken all together, as I mentioned, raised what we think is a really important question. So we wanted to determine whether hyperglycemia actually causes poor adaptations to exercise training and blunts VO2 max, especially with rising metabolic disease in the population and also consumption of Western or high-fat diet. Hyperglycemia is on the rise as well globally. So if there is some implication for the presence of metabolic disease hindering aerobic adaptations, it would be really important to address this in order to prevent not only metabolic disease, but also its connected risk to mortality. So to address this question, we used a translational approach. So in the paper, I won't present all of the data today in 20 minutes, but we used a rat model of low and high response to training, a few mouse models, some cell culture to verify the mechanisms, and we also did a small human study as well. So today, as I mentioned, I'll focus on some of the model development, in vivo outcomes, and skeletal muscle remodeling. So for the first while during my postdoc, we spent quite a bit of time developing models to address these questions. One of the challenges was that we specifically wanted to draw conclusions about hyperglycemia itself and not other confounding factors, including insulin or body weight. We wanted to focus heavily on body weight since a lot of our treadmill testing and acute exercise bouts are done on an incline in order to make the intensity appropriate. So we didn't want high-fat diet or Western diet feeding to be confounded by that factor. So what we did was start developing these models in the lab. And we actually chose to use CD1 mice. This is an outbred strain. So they're typically a little more variable than C57s, I would say, but we thought in choosing this more heterogeneous strain of mice that the translation might be a little more clear to humans, which is the ultimate goal. And so basically, just to summarize this group, they were euglycemic or with normal glycemia. They had normal circulating insulin when we measured it, and their body mass was lean. Then we introduced in a separate subset of mice a Western diet feeding paradigm, partially because, as I mentioned, consumption is increasing in the population, but components of this particular dietary regime have been shown to impair aerobic exercise capacity as well. So we fed CD1 mice, again, with Western diet. It was 43% carbohydrate and 40% fat. And not surprisingly, this led to hyperinsulinemia in this group and a slight increase in body mass, about 30%, so a little bit less than what you might expect from a straight high-fat diet paradigm. And in order to control for this body weight issue, we also implemented a second model of hyperglycemia using streptozotocin. So some of you may be familiar with this particular drug from type 1 diabetes work or immunology. It acts on the pancreas and partially reduces insulin secretion into the circulation. So we modified the dose a little bit so that it wasn't as extreme as prior studies. And this was to also avoid muscle atrophy since insulin is a growth factor. And in doing this, we optimized this model to have a slight decrease in insulin, which I'll show you on the next slide, but the body mass was the same as the control group. And we maintained them on the same diet as the controls. So it took a little while to optimize these models in the mice that we had, but we carried this forward and characterized them carefully before completing an aerobic exercise training study. So we found that at 8 weeks in some of our pilot data, this is the area under the curve for the glucose tolerance test. And so a smaller area under the curve indicates a better clearance of glucose by peripheral tissues. And we can see here that the Western diet and streptozotocin groups were significantly glucose intolerant compared to the control. And again, this was at 8 weeks. This was important to phenotype because, again, we wanted to figure out the causal nature of hyperglycemia on exercise adaptations. So we wanted to induce this phenotype prior to training them. As I mentioned as well, we wanted to be careful about body weight with these studies. So we can see the Western diet did have a slight increase in body weight compared to controls, whereas the streptozotocin group, at least in the pre-training phase, was the exact same as the control group. So this dose of streptozotocin worked really well for addressing the questions that we were interested in. I mentioned as well that the insulin profiles of these two hyperglycemic models were different. So of course, with Western diet feeding, this creates a situation of excess insulin release into circulation. And conversely, with the dose of strep that we used, it caused a partial ablation of insulin secretion. So both of these would underlie hyperglycemia, just with differing insulin profiles. And so by creating these models, it allowed us to create conclusions about hyperglycemia specifically in absence of body weight or insulin as confounding factors. So we moved forward and designed an aerobic exercise training study. So again, we used CD1 mice in these studies, and we maintained after arrival on control or with Western diet feeding or streptozotocin treatment, and we carried these forward to 8 weeks. At this point, we did baseline testing. So this included a GTT to assess their glucose tolerance or overall metabolic health. We also did an exercise capacity test at this time point. So this was on a treadmill at a 5% incline. We started them at 5 meters per minute, increased to 25, and let them run to exhaustion after that. Some people tend to use 15% or degree inclines, but again, to minimize the effective body weight, we chose less steep incline so as to not completely disadvantage the Western diet fed mice. We then allocated mice to remain sedentary in their cages, while the other half were allowed access to running wheels. So this is a quick clip from one of the mice that was in our study. You can tell that they really like the wheels. And actually, over the course of 6 weeks, they accumulated on average about 500 kilometers. Interestingly, we didn't see any change in running behavior between our control and our hyperglycemic mice, but they did run well, and they appreciate this as cage enrichment, I think. So we let them run uninterrupted for 6 weeks, and then we repeated the metabolic testing again in the form of a GTT and the same protocol for exercise capacity. Then towards the end, between the testing, we let them run for an uninterrupted period in order to preserve the training adaptations that manifested in muscle. Then at the end, we removed wheels for 24 hours in order to remove the effect of the last acute exercise bout. And during terminal collection, we collected blood, muscle, liver, and adipose tissue to assess in vivo beforehand, but on tissue-specific adaptations to the exercise training itself. And so what we found was that mice with hyperglycemia do improve their metabolic health after training, at least partially. So again, this is data from a GTT where we injected a 2 grams per kilogram glucose bolus and measured glucose over 2 hours. So we can see relative to the sedentary controls, both the Western diet and the streptozotocin groups, improved their glucose tolerance with a lower area under the curve. So this was good. We also saw other indices of improvement on the metabolic side, including a decrease in body fat. The Western diets lost some weight. We saw a decrease in adipocyte cross-sectional area and also a decrease in liver glycerol. And we also did see typical training markers that occur in response to training, including GLUT4 and an increase in many of the OXPHOS subunits. I'm not showing that today, but my point here is that wheel running did partially improve metabolic health in these mice with hyperglycemia, although not to the extent of the control group. However, as I mentioned at the beginning, our main outcome of interest was exercise capacity. So what we did here, as I said, was measure pre and post-training exercise capacity on the treadmill. And similar to the heritage study that I showed at the beginning, although with a smaller sample number, we plotted the individual improvement in exercise capacity in our groups. So on average, we have quite an interesting spread again, even in the control group. We have some that are on the low response end, but overall, on average, the ability to improve exercise capacity with training was blunted significantly in the Western diet and streptozotocin group. And we also followed up with a a test at the end of training with our time-matched sedentary controls and exercise trainers to assess VO2 peak. And what we saw here was the same thing as well. So regardless of whether we use an endurance protocol or a maximum protocol, the control animals were able to improve their capacity, whereas the hyperglycemic groups did not. So this led us to conclude that hyperglycemia is a negative regulator of improvements in exercise capacity, although the training dose was the same in these groups. So then, of course, we began to think about mechanisms and what might be underlying this phenotype of low response during hyperglycemia. And I mentioned at the beginning that VO2 max is a complex trait that's partially attributed to genetics, but it's also modifiable. This has been many years of research now to identify or delineate some of the the factors that underlie VO2 max. But we primarily understand now that it's the capacity to deliver oxygen to working muscle that underlies VO2 max. So we thought it would be interesting to look at capillary density and oxidative fiber type in these muscles. And I'll remind you again that muscle remodeling typically occurs with aerobic training. So with training in this group here, there's an increase in capillary supply for fiber, and this correlates very strongly with maximal oxygen uptake, or VO2 max. And this is again to supply oxygen to working muscle. Again, this is cross-sectional data at the bottom, but we can see in athletes that are throwers or sprinters, their fiber type tends to be glycolytic in nature. On the other hand, distance runners tend to present with an oxidative fiber phenotype. And this is specific remodeling that contributes to oxygen use and substrate provision in those contracting muscles. So when we looked at gastrocnemius muscle under the microscope in all of our models, what we see here is a healthy increase in capillaries in the control group, but this response was absent in both the Western diet and the streptozotocin animals. So this training-induced angiogenesis that normally occurs is blunted with hyperglycemia. And in line with this, when we looked at fiber typing as well, so the pink and blue are our oxidative fiber types, we see a steep increase in the control group, and this response is far less with hyperglycemia. So from the sedentary to the exercise training conditions, we don't see much of an increase. So in line with the capillary density, the training-induced shift that we expect to go from glycolytic to oxidative muscle phenotype is reduced with hyperglycemia. And so adaptations to aerobic training, as many of us are aware, arise from repeated acute signaling events at the muscle. So we tend to think about it as a change in mRNA in the hours to days after an acute exercise bout. And this will underlie translation to proteins and enzymes that are important for oxidative metabolism and many other pathways, depending on the modality of exercise. Overall, these changes at the molecular level contribute to muscle remodeling that we see depending on the exercise stimulus, and in some cases, an increase in exercise capacity. I'll also mention as well that distinct signaling events are elicited by aerobic versus resistance exercise. So of course, this figure is very simple compared to all the research that's been done in this area, but I wanted to just reiterate again that muscle contraction during aerobic activity tends to activate pathways like AMPK, calcium signaling, ERK, and these contribute to upregulation of oxidative metabolism, which underlies an increase in exercise capacity. On the other hand, when exercise is done acutely and is resistance-based, this is characterized by more high force production contractions, so a different pathway will be activated, including MAP kinase due to adhesion complexes on the membrane, downstream junk, which I have highlighted as it's coming up in the next couple of slides. And this will activate muscle growth or muscle hypertrophy in response to this training stimulus. So in prior work from the Lessard lab, we showed that in response to resistance exercise, junk or C, Jun, and terminal kinase is activated significantly with resistance exercise at both the P54 and 46 subunits. This is very obvious from this Western blot here. The immediate response to exercise is quite high. On the other hand, in response to aerobic training, junk is activated much less. So it has a low activation in response to acute aerobic exercise. So this led us to conclude that, as I mentioned, junk is activated with resistance exercise and not aerobic. In the same paper, we also looked at muscle-specific junk knockouts to assess what role junk plays in adaptations to aerobic exercise training. And again, we determined here that it's an important signaling molecule. So when junk is knocked out, we can see progression to a much more oxidative fiber type in both the fibers itself, but also the capillary density compared to the wild type mouse. So when junk is knocked out, we tend to have a more oxidative phenotype in the muscle. So with these last two slides taken together, the bottom line is that junk is a molecular switch that is activated with resistance exercise and impairs aerobic remodeling. And so since we saw this impairment in aerobic remodeling in our hyperglycemic models, we wanted to test whether there was an alteration or an exacerbation of junk signaling in our hyperglycemic models that could be explaining their phenotype. So I apologize as the next slide is a little bit busy, but I'll go through for a minute or so. Basically what we found is that when we subjected, in this case, our Western diet animals to an acute bout of exercise, the junk pathway, which we know is a negative regulator of aerobic remodeling was activated. So what we did here was run mice at a moderate speed for 30 minutes, and we kept some as a sedentary control group, and we removed gastroc immediately after to check signaling in response to acute aerobic exercise. It's probably easiest if you look at the blots in this case, but our Western diet group that exercised is on the far right side. Our sedentary controls are on the other side, and the control is beside the Western acute exercise group. And I have it highlighted here that the entire signaling pathway from junk's kinase MKK4 through junk itself and SMAD2 linker, as well as P70S6 kinase, were activated inappropriately in response to aerobic exercise. So this is kind of activation that we would expect in response to resistance exercise. And we think that it at least partially explains the failure in the hyperglycemic models to remodel aerobically after training. So, and I should also mention as well that this pattern was actually the exact same in the streptozotocin group as well. So we think it's hyperglycemia that's causing activation of this signaling pathway. I just have a couple more slides and then I should finish up. So we thought about what might be upstream of junk and causing this exacerbation of stress kinase signaling. And I don't have time to walk through all the data, I'm sorry, but what we did find in these models is that in the extracellular space that exists above the adhesion complex above junk, we see an accumulation of extracellular matrix in both the Western diet and streptozotocin surrounding the muscle fibers. So again, this is Gastrocnemius muscle, and we stained with wheat germ agglutinin, which broadly stains the matrix, and we can see a significant upregulation of the matrix in response to hyperglycemia. And other groups have shown this as well. So what we think is happening is that there's remodeling in response to hyperglycemia in the muscle extracellular matrix, which would cause stiffness or rigidity or cross-linking or just an overall increase in density, and that the adhesion complexes and mechanical signaling downstream would be exacerbated. So essentially, these hyperglycemic models are interpreting aerobic exercise as resistance, and this explains why they don't remodel their muscle in response to aerobic training. Also, we find this compartment particularly interesting because it can mediate important exercise-related processes, including angiogenesis, which we show in the paper in case you're curious, and also mechanotransduction, which I've briefly touched on here. So in conclusion, we show that hyperglycemia is a negative regulator of aerobic exercise capacity, due at least in part to matrix accumulation surrounding the fibers. We think that this causes an increase in mechanical stress and exacerbation of this stress kinase signaling pathway that promotes a glycolytic or growth phenotype in muscle. And we think that this inappropriate activation during aerobic exercise blunts typical adaptations that we see in response to aerobic training, including capillarization and a shift to an oxidative fiber type. And so we think the next step would be to determine whether reversing hyperglycemia might restore the response to aerobic exercise training. And I've got a lot of people to thank, but the Lessard Lab has been great for the last couple of years, and I appreciate the opportunity to work on this project. So I know we're short on time, but happy to take any questions. Thank you.
That was fantastic, Tara. Thank you very much for a very stimulating talk there. We've got a lot of questions which have come through the Q&A button, but I'll just kick us off for one. And Sarah, if I could direct this towards you. I noticed that you saw an increase in FGF21, and your response with your neutralizing antibody suggests that this was responsible for the improvement in glucose tolerance. Did you investigate whether this was an increase due to the activated automated stress response, so via the ATF4 pathway?
So we looked at ATF4 mRNA, and there was no difference at all in that. So FGF21 was sort of our saving grace to try and get a manuscript out, and lucky for us, reviewers didn't really poke us too hard in trying to find the exact mechanisms between the C18 ceramides and the FGF21 mechanism, but that's all we sort of looked at in response to that question. Yeah.
No, that's very informative, thank you. One of the other questions which came through on the chat section was from Declan Ryan at the University of Northampton, and he was asking, is there any available evidence to suggest how physical activity might influence the presence of these ceramides, in particularly in relation to type 2 diabetes?
So I haven't exactly kept up with exercise sort of physiology with regards to ceramides, but what I do know is that a lot of the work looking at ceramides in skeletal muscle in the early beginnings was looking at total ceramide levels. And if there was exercise paradigms included, then they definitely reduced total ceramide levels. With response to specific ceramide species now, I'm not sure. I couldn't say specifically if there has been studies or not, so I'll probably pass on that. But what is coming out of the US in the Mayo Clinic is they're actually looking at specific ceramides in the circulation as markers and predictors of cardiovascular disease and type 2 diabetes. So with that sort of respect, you would think that exercise would obviously improve your metabolic health. You should get reductions in ceramides and specific species in the circulation. As looking at these ratios of ceramides and dihydroceramides, there actually have been studies that have shown that it can predict up to 10 years in advance of whether you'll get type 2 diabetes or have a cardiovascular event, which is actually quite remarkable. And that's also been studied in mice, obviously not for the same time frame, but it's looking quite robust. And the Mayo Clinic is now pushing that out as one of its clinical tools, you would say, for cardiovascular disease and type 2 diabetes.
One of the other questions which came through, Sarah, was from Eliza Velaboas, who said that it was a very nice talk. And did you try isolating any muscle fibers and then challenging them with insulin and looking at the response in ceramides?
No, we didn't. That would probably be a really nice tool to use, I would have thought, in retrospect now. At the time, it was really trying to push to get things published. We did go through a lot of different scenarios in how we could actually try and get this stimulation sort of to mimic what we expected to see. But I think with ceramides, you expect to see something and then everything is completely reverse or unexpected in my experience of working with that and with transgenic animals.
Okay, thank you. I'll hand over to Pardeep for some more questions. Thank you, Pardeep.
Cheers, Joe. So, Tara, I have some questions for you as well. So, Tara, do you think that with a longer period of exercise training, you might have seen better remodeling in the Western diet with the mice strain that you used?
Yeah, so I'm curious about this too. The only thing that we're thinking is that there's pretty good evidence that these extracellular matrix proteins are large and very slow to turn over. So essentially, once they're modified and cross-linked and rigid, it's really hard to reverse this. So if it is the case that the matrix remodeling or density, however you'd like to call it, is imposed by the hyperglycemia, it might take a long time to reverse it, or it could be irreversible. I hope that it might be for the sake of metabolic and cardiovascular health, but we're not sure about that.
And also, Tara, so how transposable are these findings to where the human literature is at the minute?
Sure. So I didn't show the data today, but in the same paper, we actually did a small human study with 24 subjects, but we did sort of repeat the clinical association that I showed at the beginning. So in this case, we saw a very strong relationship between glucose tolerance and VO2 max in the subjects. And in those that had poor glucose tolerance, we saw an exacerbation of junk signaling. So it seems like from prior rat work that I didn't show from a few years ago to mice to humans, the mechanism seems fairly well conserved, which we were happy to see.
Thank you, Tara. So just one final quick question. So you showed some really nice, interesting mechanistic work, especially with the junk pathway there. Does this have any implications for people who are investigating any concurrent effects of doing resistance and aerobic exercise training at the same time?
Yeah, that's a good question. I think in terms of health prescription, it's still best for people to do resistance and aerobic exercise training. So I should have said actually that it's not that junk signaling is bad per se, it's just unexpected in response to an aerobic stimulus. So it could be the case that these mice are improving their muscle strength or having other benefits to training. Just if the goal is to improve your aerobic capacity and there's activation of the resistance pathway, in this case, it's kind of maladaptive. But I'm not sure exactly of how the interplay of adding resistance training might benefit health in our hyperglycemic models.
Sorry, Pardeep. I was just going to ask, the activation of the junk pathway, Tara, how long did that persist for? Do you have any idea?
So we took muscle out just after 30 minutes of running. We didn't do a time course or anything, as activation, since it's a stress kinase signaling pathway, tends to be pretty acute. I imagine it would decrease after time, but in some of the human work that we did before, junk was activated, I think 15 minutes after exercise, and then still had subtle elevation up to an hour afterwards. So the signaling pathway is still on an acute scale, but kind of drawn out to about an hour or so, at least in human work, but I've not studied this in our mice.
There was a question from Chris Holton in the chat, and he references a paper that I'm not particularly familiar with, but I'll have a go at asking the question anyway. He said there was a paper looking at 55,000 subjects, and they were showing endurance exercise 1 to 5 days a week had large reductions in all-cause mortality and reduced rates of CVD with that. However, they also show that if you had more than 5 days of exercise per week, this effect was blunted. Now, were either of you able to comment on that? As I say, it's not a paper that I'm familiar with, so.
Yeah,
I'd hand to Tara first.
I've not seen this one either, but I probably should. In this case, I'm not sure. I don't know if it's the case that there's some stress pathway that's activated, or it may be sort of simulating overtraining for relatively untrained people. I guess it would depend on the study population. Yeah, I'm not sure. Although thanks for bringing it up, because I'll have to read this afterwards. Sounds interesting. Sarah,
yeah,
I
find it interesting that 55,000 subjects stayed on doing exercise for that long. From my experience, study participants don't always adhere to protocols that are given to them, so that's quite remarkable, and it's definitely an interesting paper that I'll probably take to Journal Club next time. So thank you, Chris, for bringing it to our attention.
Sarah, there was also a question from Ian Salt in the chat who asks about your skeletal muscle specific knockouts that have reduced C18 ceramide synthesis. Were there any changes in AMPK, which might account for therefore the changes in glucose metabolism?
We didn't see any changes in phospho-AMPK or total AMPK levels. That's obviously something we also went after. I worked in the lab of Bruce Kemp for the beginning of my PhD, so I definitely tried all angles. Of course, Western blotting doesn't represent everything, but as we didn't see anything there, we tried to go after something that would give us a difference and hopefully explain a mechanism in our paper. So no, we didn't see any difference there.
Right, well, with that, I'd like to thank you both for 2 excellent talks today. It was a really enjoyable session. Thank you for all of the questions that we received as well. Next week, the seminar series will concentrate on cardiometabolic dysfunction in obesity, and I hope you can join us on Tuesday. Thank you very much, everyone.